Alrighty, well, hello everyone and welcome back. Professor Kyle here. Today we are going to be talking about blood stuff. This is going to be a fun one. We're going to talk about a variety of topics. We're going to go over hematocrit, which is going to be kind of the general composition of blood, the ratio of the different things that we find in it. We'll talk a bit about the immunological component of blood with regards to our white blood cells and the various different types of white blood cells, along with how to identify them and their kind of basic function. And we will talk a bit about blood typing and kind of the nuances of what the blood typing system is, why it's clinically relevant in terms of transfusions and the immunological reactions that can occur with that, whether it's agglutination within a patient that's receiving a blood transfusion, or potentially also like hemolytic disease of a newborn where there's a blood incompatibility between mom and child. Now, that being said, I'm going to treat this like a crash course video. So I'm going to talk as fast as I can. So if I misspeak, give me a little bit of grace. It's been a while since I've done one of these, but I finally got the whole system set back up again. So let's dive right in and get started. All right. So getting things started, if my slides will work. All right, let's just make sure we're all on the same page here, okay? So if you think back to the recesses of your mind, if you haven't compartmentalized it and gotten rid of it, we have four main categories of tissue that we learned about in AMP1, all right? Starting things off, we have epithelial tissue. This is a fairly intuitive category. This is gonna be stuff tight like your skin, all right? Your skin is an example of an epithelial tissue. Typically serves a function of sort of kind of covering or separating things, all right? So your skin is obviously separating you from your external environment, protecting you from all the nasty things that would like to devour your tissue. Next, we have connective tissue, which is where we're going to spend the majority of our time today. All right, connective tissue has a variety of things within it, and we'll talk more about that in just a moment. But blood is actually a type of connective tissue, believe it or not. Then, of course, we have muscle tissue. Remember, we had three different muscle tissue types to know. Here we have a picture, a histological picture of skeletal muscle, which is what we have voluntary control over. Then we also had cardiac tissue, which is going to be your heart muscle. And then we have smooth muscle lining kind of the lumen of your blood vessels. Think of like your arteries and that sort of stuff, as well as like the lining of your digestive system to kind of move through food through your digestive system through the process of peristalsis. Then lastly, we have nervous tissue. We're not going to talk too much about that, although nervous tissue is cool. Maybe in a future lecture we will. All right, but now we're going to focus in on connective tissue because blood is a type of connective tissue. Now within connective tissue, we have four main classes of tissue to talk about. Sorry, I'm going to say that word a lot today. We have four main categories. We have connective tissue proper, which is going to be things like your tendons and your ligaments. We're going to have cartilage, which we can think of as kind of like lining your long bones, as well as forming like the fibrocartilage of your intervertebral discs of your spine. Then, of course, we have bones. We all know what those are. And then lastly, we have blood. Now, if you look at this category, it's a fairly diverse category, and it might seem kind of arbitrary because these are very different things. Blood is kind of nothing like tendons, if you really think about it. However, there is a scientific reason for this. Quick little fun fact. During early embryological development, your tissues kind of segment into three main tissue types, okay? And then from those tissues, we kind of have the emergence of everything that makes you from a biological standpoint. All four of these categories of connective tissue, cartilage, bone, and blood all originate from the same layer called the mesenchymal layer, okay? And so these are essentially derivations of that process as they eventually differentiate into their various classes. Connective tissue is pretty cool. It's pretty broad. It serves a variety of functions, whether it's kind of support functions, structural functions, such as your bones, binding things together, which is going to be kind of your tendons and ligaments, insulating or kind of providing kind of an abrasion or an impact resistance, which is going to be cartilage, and then also sort of a transport function, which is what we're going to focus on today with regards to blood. Okay. Now, as a quick summary of that, here we can see the various categories again. So we have fibrous connective tissue, which is going to be your tendons and ligaments. We also have adipose tissue up here. Adipose is a fancy term for fat. So technically fat tissue is also a type of connective tissue, serving that sort of storage, insulation, a little bit of a protection function as well with regards to visceral fat. Linings, for example, like the female reproductive system has visceral fat kind of to insulate it from any sort of damage. We have loose areolar connective tissue. I won't talk too much about that. That's underlying your skin and your skin epithelia, cartilage, which we've already mentioned, bone, which we've already mentioned. And lastly, we have blood, which we're going to talk about more today. All right, now, what is the composition of blood? So this is going to be our discussion of hematocrit, if this is relevant to you in your coursework. We have two broad categories for hematocrit. We have blood plasm, or plasma, and then we have formed elements within our blood. And formed elements is going to be where all of our cells reside. That's going to be that category. And the plasma is going to be kind of everything else. It's going to be water and then various forms of solutes within that as well. Now, speaking of blood plasma, so blood plasma typically on average is going to sit somewhere around 55%, 50 to 55% of your total blood volume is going to be the blood plasma. The majority of that component, so of that 55%, somewhere in the range of about 90 to 95, averaging somewhere around that 92, 93% is actually just going to be straight up water. Okay, so the vast majority of your blood plasma is going to be water. That's why it's important to stay hydrated in order to maintain blood pressure, because if you get dehydrated, this proportion is going to go down, which is going to decrease your total blood volume, decrease your blood pressure, potentially cause you to pass out if you stand up too quickly. Okay. And this is very useful because it adds a lot of volume. It makes the fluid much more liquid, much more less viscous. We'll talk more about that in a little bit. And it provides kind of the solution that we can dissolve everything else and that all of our cells can live within. Speaking of solutes, we have these things called salts or electrolytes. So electrolytes is basically just the fancy scientific term for salt. So if you've ever had like a sports drink, like a Gatorade or something like that, and it says it has electrolytes in it, that's basically just a fancy way of saying it has salts in it. So these are going to be things such as like sodium, potassium, calcium, and the list goes on. These serve various functions for the purposes of maintaining kind of our blood volume. They help maintain osmotic balance or osmotic pressure. I'll mention that again in a moment. And also they serve a metabolic function with regards to kind of communication within your muscular tissue. So again, we utilize these little ions, these little positively charged salt ions, to kind of help activate the process of neuromuscular activation. So we can trigger that activation cascade to allow for synapses to fire and for muscles to contract. Okay. Then we have this category down here of plasma proteins, blood plasma proteins. Couple important ones to mention, may be relevant to you, maybe not. We have albumin. Albumin does a couple different things. It's very important for maintaining that osmotic gradient. To kind of explain that a little bit further, osmotic pressure or osmotic gradient is basically just, if you think back to your early chemistry classes in high school, again, water is going to try to maintain its balance across a semi-permeable membrane. And so if we have things like salts and proteins dissolved within our blood plasma, that's going to pull water into the lumen of the blood vessel and increase our blood pressure, all right? Whereas if we need to decrease our blood pressure, we can get rid of them and excrete them, for example, through our urine when they get kind of travel across the membranes of our kidneys and that sort of things. Albumin's also useful as kind of like a carrier molecule, so it helps transport other things that don't tend to dissolve very well into water, and so they can function in that regard as well. Fibrinogen is going to be important for some stuff that we'll talk about later with regards to coagulation, so blood clotting. Fibrinogen is turned by an enzyme called thrombin into fibrin, and fibrin is going to be one of the main molecules that makes things sticky within your blood so that we can help kind of clump things together and trigger that process of coagulation. And then there's globulins. We don't need to talk too much about that. And anything else we need to talk about with plasma? No. Again, I'm trying to do this relatively quickly. I know you guys usually watch my lectures at like 1.5 times speed. I'm trying to talk at 1.5 times speed to start. So here we go. Excuse me. All right. Now, with regards to formed elements, all right, we have a couple of different things to note in formed elements. We have our erythrocytes, which are going to be our red blood cells. These are what we typically think of when we think of blood. Okay. This is what gives it the color. And the main function of blood, or one of the main functions of blood, is to be providing oxygen to our various tissues for the purposes of aerobic respiration, which is how we produce energy. We produce ATP, which is the most fundamental molecule for kind of accomplishing energetic functions, metabolic functions within our cells. And the most efficient process for that is aerobic respiration, which we need oxygen for. So we need lots of oxygen. Erythrocytes are very good at carrying that. A byproduct of respiration is going to be CO2, and CO2 we need to get rid of. We don't want too much of that in our blood or in our body in general. And so when red blood cells reach their target tissue, they're going to drop off the oxygen. They're going to pick up the CO2, bring that back to our lungs, and then we're going to have that process of gas exchange within the alveoli of our lungs, where again we drop off the CO2 that we've picked up from our body, pick up oxygen from the air, and then the cycle repeats. Okay, we'll talk more about red blood cells in a little bit. Next, we have white blood cells, or our leukocytes, as they're more scientifically known. So leukocytes is the scientific name for our white blood cells. There's 5 main white blood cells. We'll talk about each of those in just a little bit. And then lastly, our third category over here in terms of our formed elements are going to be these things called platelets. Platelets are basically just tiny little fragments of cells. The cells that they come from are called megakaryocytes. You might need to know that term. And megakaryocytes are very large cells, and when they've reached maturity, they'll commit apoptosis. They'll break off into tiny little pieces, and these tiny little pieces form the platelets, and the platelets are going to be the main part that, or an important part rather, not necessarily the main, but important part of that process of coagulation so that we can stop blood flow from an artery that's been damaged or lacerated, okay? So that's going to be it there. Now, in terms of hematocrit and overall blood volume, kind of what are some of the important facts to know about here? So again, we already mentioned that the typical proportion is going to be plasma is going to make up about 55% of your total blood volume, and the formed elements, predominantly red blood cells, is going to be about 45%. An interesting distinction between males and females, both the proportion of the hematocrit, so the proportion of red blood cells that they have, as well as the total amount of blood that an individual has, will vary a bit based off of sex. So adult males are typically on average going to have between 5 to 6 liters of blood, and females are going to have between 4 to 5 liters of blood. This is basically just a consequence of the typical size of difference between males and females. Males tend to be a bit larger, therefore they have proportionally more blood. All right, nothing too crazy right there. Anything I need to talk about on this slide? No, this basically just further explains what I just discussed. Okay. Again, just to further break down within blood plasma, what are some things to kind of take note of that are dissolved within that? Most of it's going to be water, and then dissolved within that water, we're going to have various proteins like albumin, fibrinogen, which I mentioned earlier. We're going to have those electrolytes, which again is just a fancy term for salt. We're going to have nitrogenous substances. Some of these are metabolic waste products that we end up excreting through urine. Others serve actual metabolic functions. We're going to have nutrients such as sugar molecules are going to be dissolved within our blood to be sent off to our, for example, our muscle tissue, which are fairly hungry tissue types because they're constantly contracting and doing work. We will have some gases dissolved within there as well. We have a tiny amount of oxygen that can be dissolved within the blood plasma, where we can also have a bit of CO2 in there as well. Typically, it turns into bicarbonate. And then we'll have various other chemical messengers. So these will be things like hormone molecules. Think of like testosterone and estrogen as like your primary sex hormones. They can be dissolved within your blood plasma, as well as some other things like cytokines and that sort of stuff, which are tiny little chemical messenger molecules. Some cytokines serve a very important function within our immune system. Okay. Now, with regards to our formed elements, again, our formed elements are going to be our cell stuff within our blood. We have the erythrocytes, our red blood cells, which are going to be the vast majority of the formed elements. And then we're going to have our leukocytes, which are our white blood cells. All right. And we have 5 white blood cells to note, and we'll talk about each of these in turn, as well as like how to distinguish them between one another. And as a useful mnemonic with regards to remembering the white blood cells, we have this mnemonic up here. This is a fairly well-known one. It's used all over the English-speaking world, whether it's the UK, America, where I'm at, or even Australia. And in fact, I learned this mnemonic in an Australian accent. I'm going to butcher it horribly, but it was never let monkeys eat bananas, okay? That's just because I listened to an old science podcast, and it was a couple of Australian PhDs, so I learned it in an accent. But never let monkeys eat bananas. All right, why is this useful? Well, first of all, as a mnemonic, the first letter is going to delineate each of the types of white blood cells that we need to know. And the other useful thing about this mnemonic is that it's going to have them in order of prevalence, as it says right here, okay? Order of prevalence. So never refers to our neutrophils. Those are going to be the most common white blood cells. And then bananas is going to refer to our basophils right here, which are going to be our least common white blood cell type, all right? And lastly, again, we have platelets, which are involved with blood clotting. Here's just a summary slide. It may or may not come out on video. I haven't double-checked the footage. I'm not going to spend too much time here. If you want to, like, look at it, I can try to get... Nope, that's not going to work because the camera's going to follow me. Anyways, this is a good summary slide. I'm not going to spend too much time here because we'll talk about each of these things in turn. Let's talk about red blood cells. All right, red blood cells, their primary job is to transport oxygen, first and foremost. All right, and then in the process, they're also going to be picking up CO2 once they've delivered their oxygen carrying capacity. Now, if you look at this cell, you may notice something interesting about it. It's something missing. All right, that's your hint. 3, 2, 1. It's missing a nucleus. All right, so red blood cells are anucleated. In other words, they have no nucleus. Why do they not have a nucleus? Well, a nucleus takes up space within side of the cell, and this guy's number one job is to transport oxygen. It does that on hemoglobin molecules, a little substructure of 4 hemoglobin subunits, at the center of which is typically an iron atom, and that iron atom is what's going to bind with the oxygen. And since their primary job is as a transport transport carrier with those hemoglobin molecules, if we get rid of our nucleus, we increase the volume of space for hemoglobin molecules, which increases our oxygen carrying capacity and just makes our body essentially more efficient. So a immature red blood cell, an immature erythrocyte will have a nucleus, but once it's reached maturity, it will dissolve its nucleus to increase room for hemoglobin. Now, that being said, an interesting fact about red blood cells is they essentially have a hard stop in terms of their lifespan of about 120 days. All right, so that's an important number to remember. I would try to maybe write that one down. But the typical lifespan of a red blood cell is going to be 120 days, and that's essentially a hard stop because they have no nucleus. So the nucleus, again, contains all of the genetic information for the cell necessary to produce all of its various components, its organelles, and that sort of stuff. And if we dissolve that nucleus, sure, we increase our oxygen carrying capacity. We have more room for hemoglobin. But now we essentially have no mechanism to repair ourselves. We can't produce any new organelles. We can't repair any of our cellular structures. We can't repair our cell cytoplasm. And so since we lose that mechanism, they essentially can't live longer than about 120 days. Our liver's very much involved with kind of filtering out our bad red blood cells. As red blood cells get older, and they start to break down, they start to get stiff, they become less flexible, and they essentially go through this mechanical process of filtration where they have to squeeze through these tiny little pores. And if they get old, then they get stiff and they can't make it through the pore, and then that's how your body is able to identify which red blood cells it needs to get rid of and which ones are okay to keep. In terms of a proportionality relative to our white blood cells, they typically, on average, will outnumber them of about 1,000 to 1. Okay, so we'll have 1,000 red blood cells to every white blood cell. This ratio can move around depending on the circumstances of the individual. Endurance athletes might have a little bit of an elevated ratio. Okay, they might have more red blood cells. We'll talk about that a bit later when we calculate hematocrit. Or if you're actively fighting in an infection, you might have more white blood cells. Or if you have blood cancer like leukemia, which is a cancer of our white blood cells, this ratio might get thrown off. But under normal circumstances, it should be roughly about 1,000 to 1. Now down here, I'll note this little bullet point right here. Major contributing factor to blood viscosity. What does that mean? All right, so viscosity is a term that describes essentially the thickness of a fluid. So if it's more viscous, if it has a higher viscosity, that means that the fluid is thicker. All right? It's approaching that end of the spectrum that's approaching like a gel. Think of like strawberry jam or something like that. It's a very thick sort of fluid structure. On the other end of the spectrum, we essentially have pure water, which is going to be very low in terms of viscosity. It's going to have a low viscosity, which means it's a very thin fluid, very easy to move through small pores. So in terms of viscosity, why is this clinically relevant? Well, it's relevant for our blood pressure, okay? So if we have a thicker blood, if we have a higher degree of viscosity, it's going to be harder for our blood to move through our small pores of our capillary beds and that sort of stuff, which is going to cause our heart to have to contract harder in order to overcome that. In other words, our blood pressure is going to be increased. So if we have more red blood cells, we have a higher viscosity, we have thicker blood. And the inverse is also true. All right, now let's talk about our white blood cells. So we have, again, 5 white blood cells to note. Never let monkeys eat bananas. And within our white blood cells, we can define them or segment them into 2 categories. We have our granulocytes and we have our agranulocytes. And that's essentially just a description of kind of the visualization of the cell, okay? So if we look right here, here we have a granulocyte with these tiny little, notice how it's kind of a little bit of a discolored function right here. We have these tiny little granules, these little vesicles inside of the cytoplasm of the cell. And for the immune cells, these are typically going to be like things such as like peroxisomes, lysosomes, and other sorts of little vesicles that contain things like enzymes and acids that are very good at breaking things down. In this case, for an immunological function, these little vesicles can be released either within the cell cytoplasm. If it phagocytizes, if it envelops a bacteria, it can dissolve it inside of the cell, or it can also release them into the external environment. It can secrete some of these vesicles to kind of poke holes in the pores of larger organisms, such as parasites, if we're fighting a parasitic infection, okay? So we can segment white blood cells into 2 categories based off whether or not they have granules, granulocytes and agranulocytes. A note right here, it mentions the stains. This may or may not be relevant to you. A quick summary note right there. Methyl blue tends to bind to nucleotides. So it's going to bind to our nucleus, which contains our DNA, which is going to be this darker, more purpley, sometimes even bluish color hue right here. So that's going to be a methane blue stain. And then eosin is a very common counter stain. Eosin is going to bind to proteins prevalently. And so obviously the majority of our cellular machinery, the stuff floating out in our cell cytoplasm, is going to be protein based. And so hence why this has a bit of a reddish stain right here. So methyl blue blue is going to be more of a purpley stain attached to nucleotides, hence it's binding to the nucleus. Eosin is going to attach to proteins, hence it's binding to stuff inside of the cell cytoplasm. Okay. Now, let's talk about each of our white blood cells. We'll talk about the basic function as well as kind of some notes to help distinguish between the various different types. So here we have a neutrophil, or the N in our mnemonic of never let monkeys eat bananas. So N being the first letter in that mnemonic means this is going to be the most prevalent of our white blood cells. So we have more neutrophils than we have of anything else under normal circumstances. Sometimes that ratio can change. We'll talk a little bit about differential white blood cell counts and why that's clinically relevant later once we get through like the individual cell types. So neutrophils are going to be the most common, and in terms of their function, they're fairly adaptable. They can attack a lot of different infections, so they'll fight bacterial infections, viral infections, to a limited extent, even some parasitic ones, potentially even cancer. So they have specific granules. The granules, I will say, on neutrophils tend to be less visually noticeable than the two other granulocytes that we'll talk about in just a moment. That being said, though, a very distinguishing feature of these guys is they're going to be multilobular. Okay, so they have this multilobular nucleus right here. So this is still just one nucleus. However, it's kind of segmented itself off into these little lobules right here. Fun fact about these, they're also useful as a metric. If we're looking at a particular cell, we can kind of tell how old it is based off how many lobes that it has. So younger ones will have 2 to 3, more mature or older neutrophils will have like 4 to 5. Typically, you don't see more than 5. So they're going to have this elongated multilobular nucleus, okay? And they'll be somewhat grainy. Maybe not super grainy, but somewhat grainy. All right, next in our granulocytes, we have our eosinophils. The take-home message for these guys is eosinophils are very effective at fighting parasitic infections. They're relatively large cells. They're going to have very much a reddish hue, as we can see right here. Okay, they have a reddish hue, and they're going to have very clear granules in their cell cytoplasm. Those are going to be our eosinophils. It also mentions here that they're involved with chronic inflammation and allergic reactions. So, like if you touch like poison ivy, eosinophils and basophils might freak out and release histamine and cytokines and that sort of stuff. And in chronic inflammation, oftentimes this is going to be in conjunction with like autoimmune conditions where we're dealing with sort of self-reactivity. Eosinophils can sometimes be implicated in those sorts of situations if an individual is dealing with chronic inflammation. Okay, but take-home message: they're good at fighting parasitic infections, and they're going to be granulocytes, so they're going to have these little granules, and they're going to have a reddish hue to them. Next and last for our granulocytes, we have the basophil. Thinking back to our mnemonic, never let monkeys eat bananas. Bananas is the last term, so these are going to be the least common under normal circumstances of our white blood cells. And basophils, they're not so good at fighting things directly, but they do serve a very important function in terms of elevating and activating our immune system. Okay, so they bind IgE antibodies. IgE is a type of antibody produced by your adaptive immune system, kind of in response to infections that you've seen before. So they're going to help kind of transition if we have an initial infection. They're going to help activate our adaptive immune system. So right now with our white blood cells, we're talking about our innate immune system. These are the guys that are always there. They're always on standby, and they're fairly effective at identifying a wide variety of potential pathogens in our body. Whereas our adaptive immune system is going to be focused in on things that we've seen before that we really want to get rid of really quickly. So basophils kind of help notify our immune system that we have a problem in a particular area and recruit more cells to the region. Part of that recruitment process is going to be this function of releasing vasoactive or vasodilatory agents. So these are going to be things such as chemicals like histamine, which is a fairly potent vasodilator. If you've ever been stung by a bee, within bee venom, there's histamine, and histamine will dilate blood vessels. So that's why you might get a little bump on your skin when you get stung by a bee. And they'll also release things called cytokines, which are little chemical molecules that create little trails that other immune cells can follow to kind of reach the area where we have an active infection. Okay. Now, in terms of identification, these are fairly easy to identify. These guys are going to have a relatively purplish hue. So if we juxtapose this to our eosinophil, again, we had more of a reddish hue right here. We're picking up a lot of that eosin dye. Over here with the basophils, we're going to have a little bit more of a purplish hue with some very clear granulocytes in here as well. It will also oftentimes be multilobular in terms of the nucleus, but it's going to have a very dominantly purplish hue with very clear granulocytes. Okay, that's going to be our basophil. Now we'll switch over and talk about our agranulocytes. We just have two to note right here. First off is going to be our lymphocytes. So never let monkeys eat bananas. Let is our next letter. Lymphocytes, as a consequence, are going to be our second most common under normal circumstances. Similar to neutrophils, lymphocytes are fairly adaptable. They can attack a variety of different pathogens, whether it's bacteria, viruses, or parasites. They're also very phagocytic, so they like to envelop things into their cell cytoplasm. They tend to be fairly large cells. Okay, they can vary in size, but the larger ones are very good at that process of phagocytosis, where it envelops things like bacteria to break them down internally in a controlled process. They can do this a number of times on in many cases. Eventually, they reach their limit, at which point they'll commit apoptosis and they'll do a controlled breakdown of their internal machinery to kind of inactivate it and prevent the internal composition of the cell from spilling out, potentially damaging elements into the blood plasm or whatever tissue they're residing in. Now, in terms of identification, how do we identify these cells? Well, these guys are going to have a very large nucleus, okay, and it's going to be a relatively round nucleus. Let's see if I can zoom in here on these guys right here. I'll look at this cell in particular. So they will have a relatively small proportion of cell cytoplasm, so they'll have a small halo of a cytoplasm, as we can see right here, kind of lining the periphery of the nucleus. And the nucleus is going to be very dominant and very large within the cell itself, okay? And notice we don't really have granules in that little bit of cytoplasm that we can see. So again, tend to not be grainy and tend to have a small halo of a cytoplasm and tend to have a very large nucleus. That's going to be a lymphocyte. Juxtapose that to our monocyte, which is going to be our last type of white blood cell to identify. Our monocytes are going to have, from an identification standpoint, they're still going to have relatively minimal amount of cell cytoplasm around the periphery of the nucleus. However, the nucleus in this case, instead of being very rounded in shape, the nuclei of monocytes tend to have this sort of horseshoe or a U-type shape to it, okay? So that's going to be the most useful distinguishing feature between the two. If you see a fairly large cell with a very large nucleus and you're trying to figure out, okay, is this a monocyte or a lymphocyte? Look at the shape of the nucleus. If it's got a U shape, it's probably going to be a monocyte. Now, an interesting note about monocyte is the name for the immature version of this cell. So monocytes will float out in your blood plasma and in sort of an inactive state. And then let's say you're now battling an infection. Monocytes will migrate to the area of an infection. Basophils are helpful for that process with the cytokines that they release. They'll migrate into a tissue, and once they reach that target tissue where they're going to be fighting the infection, they go from being called a monocyte to a macrophage. And a macrophage, again, very large cell and very phagocytic, all right? They're very good at enveloping other cells, such as bacterial infections, and activating them that way by breaking them down internally within themselves. So in terms of identification, they're going to be a fairly chunky boy. They're very large cells because, again, they turn into macrophages that are going to devour other things, and they're going to have sort of a U-shaped nucleus, all right? So that's going to be it for our white blood cells. Lastly, in terms of identification of sort of our formed elements, so we talked about our red blood cells, we talked about our white blood cells. That third category on that earlier slide we saw was platelets. And what are platelets? Well, platelets are going to be these small fragments of a cell type called a megakaryocyte. That we can see that term right here. If spelling is important, go ahead and take a mental picture of that term right there, megakaryocyte. So megakaryocytes are fairly large cells, and then once they reach maturity, they'll break down, they'll commit apoptosis, and then the fragments of those cells will form these little platelets that essentially just hang out inside of your blood plasma. And when necessary, they will be a very important part of the process of coagulation. Think of them as kind of like a bit of a glue to the matrix of a blood clot, okay? So these guys will get sticky. They'll start attaching to each other, attaching to red blood cells, and they'll initiate that process of coagulation to eventually occlude blood flow, stop blood flow through the affected blood vessel or artery, okay? Here's a nice summary slide where we can see everything that we just talked about. So again, erythrocytes, or our red blood cells, are primarily involved in the transport of gases such as oxygen and carbon dioxide, delivering oxygen to our tissues and picking up CO2 that we want to get rid of. Neutrophils, the most common white blood cell, are going to be involved with phagocytosis and breaking down pathogens that we encounter. Eosinophils, also involved with that process, very good at fighting parasitic infections. Basophils, kind of notifying the immune system that we have an infection and recruiting more cells to the area. and causing dilation in that region with like things like histamine and that sort of stuff. Lymphocytes also going to be fighting infection, and fairly adaptable in that process. Monocytes are going to turn into macrophages when they get activated and fight infections. And then thrombocytes or thrombocytic fragments are going to be involved with blood clotting and kind of the reparative process once we get to the tail end of dealing with like a laceration to a blood vessel. Okay. Here's a nice summary slide where we can see all of this stuff. So again, to go through a quick process of identification, we'll just kind of run down this list. Again, red blood cells are very easy to identify. They're going to look different than every other cell, especially because they're anucleated. They don't have a nucleus anymore. Then we're going to have our neutrophils. Our neutrophils are going to be multilobular with their nucleus, and they're going to have small little granules inside of them. Our eosinophils, I can't use my side note. I can't use my laser pointer when I'm zoomed in. So just kind of visually track with me as I can't reach that far into the frame. It's going to cut off. Yeah. Okay. Eosinophils are going to have a reddish hue to them. Large cells with a large nucleus, very grainy, and they'll have a reddish hue. Basophils, juxtapose those two. Basophils are going to be very grainy, but they're going to have more of a bluish purple hue to them. Lymphocytes are going to be cells that have a large nucleus with a small amount of cytoplasm, but they're going to have rounded nuclei. And then lastly, our monocytes are going to be very large cells with a sort of a U-shaped nucleus to them. And then lastly, platelets are those little bits of fragmented cells floating out in the blood plasma. All right. So that's a quick summary right there. Here's a little review exercise that I would normally use in class, like a little pop quiz, if you will. It's not graded, but it's a fun little exercise. This is a recording, so it's not useful for you guys. So if you want to go ahead and test yourself, go ahead and pause. 3, 2, 1. All right. And then here are the answers. So again, going through that process, neutrophil, multilobular nucleus, little granules inside of it. That's going to be a neutrophil. Lymphocyte is going to have a very large rounded nuclei with a small halo of cytoplasm around it. Monocyte is going to be a very large cell with a large nucleus. However, it's going to have a U shape to that nucleus. Eosinophil, granulocyte with a red hue, and then basophil, kind of a purpley blue hue to it with granules. All right. Blood smear, I'm just going to kind of glance over. This is a thing sometimes done. Maybe your school does it. I don't know. Our school doesn't do this anymore, so I'm just going to skip over those. All right, hematocrit. We already covered some of this stuff. Just a quick review, and we'll kind of add a little bit more color to some of these things. Previously, we mentioned that one of the differences between males and females is the total amount of blood volume that an individual will have, essentially just as a consequence of their size. So larger individuals, males tend to be larger. So therefore, they'll typically have on average between 5 to 6 liters of blood in circulation at any given time. Females will have between 4 to 5 liters of blood at any given time. Another difference is actually going to be the composition or the proportion of red blood cells to their total blood volume. So not just the total amount of blood that they have, but kind of the proportionality of it. Now, here on this slide, we can see some clarifications on that. Males tend to, on average, have a bit of a higher proportion of a hematocrit. So they'll have more red blood cells on average, typically floating somewhere in the range of about 47% of their blood volume, again, plus or minus 7. So there's a range where this can occupy. Females on average will have slightly lower hematocrit, so somewhere around a proportion of 42% of their total blood will be red blood cells, okay? Now, in terms of calculating these percentages, it's a very simple bit of math. I'll kind of I'll scoop my head over here. Improvise, right? It's very simple math, okay? Yes, there's a little bit of math, but it's as simple as it gets. It's just a ratio of what the proportion of red blood cells are to the total volume of blood. So if we look at this right here, this little number over here on the side. All right, so if we want to calculate our hematocrit or our proportion of red blood cells to total blood volume, we'll take the proportion of this vial that is occupied by our red blood cells. In this case, we're going up to about the 6.5 mark right here on this vial, whereas our total blood volume in this vial is going to be about 13 centimeters. So if we do 6.5 over 13, that's a perfect 1.5, or 1/2 rather, which will be a hematocrit of about 50%. Okay, so very simple math. Don't get too scared by it. Another interesting note: I'm something of an endurance athlete, and when I was, can I get the camera to move? Thank you. Back in the day, so I use a lot of endurance examples. I was something of a cyclist when I was cool. So one of the things that can happen as a physiological adaptation to endurance training is your hematocrit may be elevated. Okay, so Endurance athletes are obviously utilizing more oxygen for extended periods of time. So one of the ways the body adapts to that is it will produce more red blood cells, so it increases your oxygen carrying capacity. It releases a hormone called EPO in order to accomplish that. EPO is the hormone that regulates red blood cell production. So you'll have elevated EPO levels, which will then lead to elevated red blood cell counts. Okay. Now, an interesting note with that. So sometimes the question is posed, well, if red blood cells are so great, why don't we just have a ton of red blood cells and relatively low blood plasma? That circles back to that earlier note. If we remember that summary graph, that last bullet point on the previous slide mentioned blood viscosity, okay, and how red blood cells are a major contributor to our overall blood viscosity. So if you have more red blood cells, you have an increased oxygen carrying capacity, which is good if you're constantly using oxygen. For example, if you're running, cycling, or swimming. However, as a consequence of that, we increase our blood viscosity, which may cause our heart to have to work harder in order to push blood through our vasculature, especially at the small capillary beds where the actual process of dropping off oxygen and picking up CO2 occurs. So if we increase our hematocrit, we increase our viscosity, which forces our heart to work harder. In the short term, that might not be a problem, but if we sustain that for an extended period of time, we could potentially cause damage to our vasculature, to the lumen of our blood vessels themselves, from constantly experiencing that increased pressure. And also it can cause some pathologies in the cardiac tissue of our heart itself. Okay. So that's why typically a normal person should sit somewhere within this range of like 37 to 54. If you go much over that 54%, your viscosity will get a bit too high and it can cause some issues. Now, if you were to do this in a lab, if you wanted to calculate hematocrit, you would take a small sample of blood and you would stick that into this little vial that we can see right here. Move my head over to the side. In this little vial right here. And then you would take that vial and you would place it in a thing called a centrifuge. And this centrifuge is a device that's just going to spin really, really fast. Think of centrifugal force if you've covered physics before. And when it spins, it's going to apply a g-force. It's going to kind of create an artificial gravity, if you will, within that space. And heavier, denser, more important factor is density. But heavier and denser things are going to go to the bottom of the vial, and lighter, less dense things are going to float at the top. So our formed elements of our blood cells are going to obviously form down here. They're going to collect down here at the bottom of this vial. And then everything that's light, such as water and kind of the solutes, think of our salts and our proteins, are going to float up here in the plasma up here. Okay. And so this is a way that you can calculate hematocrit, just taking that proportion that we mentioned right here with this calculation over on the side. Okie dokie. Bleeding time. All right, I won't spend too much time on this in the recorded lecture. I sometimes bore students with my examples in the classroom. Bleeding time on average is going to sit somewhere around 5 minutes. 2 to 7 is going to be relatively the normal range, okay? If you're in excess of that, then you might have some other issues going on. You might have potentially a deficiency in one of the clotting factors, one of the molecules involved with triggering coagulation. But typically, on average, an individual will have a bleeding time of about 5 minutes. So it takes about 5 minutes for the coagulation process to be activated sufficiently in order to occlude blood flow through the vessels that have been lacerated and are actively bleeding. That coagulation cascade you may cover in your more advanced classes. Suffice it to say right now that some important molecules involved in that is going to be the fibrinogen that we mentioned way at the beginning of this lecture. So fibrinogen is going to be activated by thrombin, which we can kind of see over here. Okay, this process over here. Prothrombin turns into thrombin. Thrombin activates fibrinogen, turns it into fibrin. Fibrin is very sticky, initiates that process of the coagulation cascade that we can see right here. Okay. Okay, now speaking of the coagulation cascade, so here we can see kind of a breakdown of the various different stages of it. You'll see these things referred to with different terms depending on what study resource you're utilizing. So, here we have like platelet phase, coagulation phase. You'll also see these referred to as like, primary hemostasis, secondary hemostasis, those sorts of things. I'm not going to get too into the weeds with the details of the coagulation cascade, at least not for the purposes of this video and for this class being AMP2. You might dig into this more in your more advanced classes. So there's some other terms that might be useful to just kind of get familiar with. So we have this thing called von Willebrand factor. And so, it's a necessary component of attachment for some of the initial platelets when they are attaching to the lining of the vascular wall. Then we have some other things like TXA2 which is thromboxane A2, and again another molecule important for the process of platelet aggregation. Um, I'm not going to get too much more into the weeds with that. Some other terms that you may want to familiarize yourself with, at least for the purposes of advanced courses, are going to be things such as like the intrinsic and extrinsic pathway, which are mentioned right here. So, extrinsic system refers to clotting as a result of kind of trauma, so damage to tissue externally, hence its name. Whereas, like the intrinsic system has to do more with just if damage is detected within the lining of the vascular wall itself, and it relies on some marker molecules that are situated on the lining of the vascular wall and that sort of stuff. And that pertains then to measuring PT time and PTT time. These are different methods for essentially evaluating the efficacy and how quickly coagulation will occur based off of the which pathway is being utilized at a particular time. That being said, 2 to 7 minutes on the prior slide. Knowing thrombin is important for activating fibrinogen and turning it into fibrin to get into like the coagulation phase is about as far as we're going to go with the details on this. Okay, so now let's talk about blood typing. What is blood typing? Why is it relevant? Why do we care? All right, so we're going to define a couple of terms here. And what this is clinically relevant for is the process of blood transfusion. So we just previously covered the process of coagulation, where we're trying to occlude blood flow if we have a laceration of an artery or something like that. Let's say we have a patient who's had a fairly traumatic injury and has lost a lot of blood. So we may need to now give them blood or give them a blood transfusion. There's a couple different types of blood transfusions that we can give a patient. One form is just whole blood. All right, so that's going to be blood plasma as well as red blood cells. Another slightly more common process, actually considerably more common process, is just to give packed red blood cells, which is basically we centrifuge down and we separate out the red blood cells, and then we give them injection of just pure red blood cells, no blood plasma. And in the case of blood typing, what we want to avoid, where's my laser pointer? Hold on. Sorry, technical difficulty. All right. In the case of blood transfusions, what we want to avoid is an adverse reaction called agglutination, which is defined over here on this next slide. Okay, so blood transfusion recipient and donors must be matched in order to avoid agglutination. Agglutination is a process that's very similar to coagulation in the sense that it's the clumping of red blood cells, but this is not an appropriate response. So in the case of coagulation, we want to have that when we have a laceration of a blood vessel. We want to occlude blood flow through that particular vessel. However, in the process of agglutination, this is just going to happen randomly throughout the blood wherever the antibodies and antigens interact, and we'll talk more about those in just a second. And so this can occur randomly, and it can lead to occlusion of blood flow to major arteries, causing infarction of this tissue that's downstream of that because it's not getting oxygen, so the tissue will start to die. These things can also migrate through the lumen of blood vessels, potentially going to some major arteries like your coronary arteries on your heart, causing either like a transient ischemic attack or a full-blown myocardial infarction, aka a heart attack. So we want to avoid this sort of clumping that can arise as a result of agglutination. Now, how does agglutination occur? Well, it arises as a result of an interaction between antigens and antibodies. Now, you'll learn more about antigens in some of your other classes, but suffice it to say that these antigens are these little markers that exist on the surface of your cells, okay? These little molecular markers. And they serve a variety of different functions. Oftentimes, they're kind of informing the adjacent cells and neighbors as to like, hey, this is what type of cell I am. This is also what I'm doing. Sometimes it's, your cells will switch out their antigens based off their particular function at a given point in time. And also it provides kind of a useful landmarker for like a postal service of the body, kind of deciding where different vesicles are supposed to bind to and that sort of thing. Now, in the case of blood typing, we have these antigens called A and B antigens. We'll also talk about Rh in just a little bit, Rh or the D antigen, which is inherited in a different pattern than what we're talking about with this table right here. And so these A antigens just exist on the surface of your red blood cells. And according to your own immune system, these indicate that, hey, I'm you, everything's good here, no need to react with me, all right? I'm not a pathogen in your bloodstream. What's interesting about these blood typing relationships is if you have type A blood, that means you have A antigens on your surface. But what your immune system will naturally do is it will produce anti-B antibodies. And these antibodies, if you think back to your introduction to kind of infection and immunology and that sort of stuff, antibodies are these little molecular markers that it float out inside of your blood plasma. And they're essentially just on standby to interact with their corresponding molecular marker, typically that of some form of pathogen. So you have your innate immune system, which was the white blood cells we talked about earlier. Then we have your adaptive immune system, which is going to be kind of maintaining a memory of previous infections, whether bacterial or viral and that sort of stuff, even parasitic. And your body's adaptive immune system will have these specialized cells that will produce antibodies with a corresponding active site right here, kind of like the little V shape in the Y of this little antibody, that corresponds with a very specific molecular marker on one of those pathogenic cells. All right. And so if they end up interacting with one of those, then that's a notification that, hey, we're fighting an immune infection and we need to recruit some more cells to fight this infection. Now, again, with blood typing, if we have type A antigens on our surface, we'll naturally produce anti-B antibodies that will float out in our circulation. And as a closed system, we don't have an issue right here because these antibodies aren't going to react with your own antigens, and therefore we won't have that process of agglutination. However, circling back to what we discussed earlier with, bleeding and all that sort of stuff, let's say we have a patient who's had a fairly traumatic injury and has suffered a significant amount of blood loss. We may need to provide them with a blood transfusion, all right, to kind of bring them back to a state of homeostasis. There's a couple different types of blood transfusions that we can give. There's whole blood transfusions, which, as the name implies, is both the blood plasma as well as red blood cells, hence the name whole blood. There's also packed red blood cells. So we can give a patient packed red blood cells, which is no blood plasma, just the red blood cells, so we can try to get them more sufficiently oxygenated. Now, again, as a closed system, we don't have an issue here because these antigens aren't going to react with these antibodies. They don't correspond with each other. But if we're giving a patient a blood transfusion, we need to make sure that we're not going to have an adverse interaction between the recipient, the patient's blood type, and that of whatever donor blood that we are giving them. So again, there's that relationship of if we have the A antigen on our surface, we'll naturally produce anti-B antibodies in our blood plasma circulation. The opposite is going to be true for B-type blood. What does B-type blood mean? Well, it means we have B antigens on the surface of our red blood cells, and that person's immune system will naturally produce anti-A antibodies that'll float out in circulation. As a closed system, no issues, but if we give a blood transfusion between these two individuals, we're going to have an adverse interaction where the antibodies of the receiving patient are going to interact with the antigens on the surface of the donor blood and cause that process of agglutination, which is what we're trying to avoid. Now, to quickly define these last two categories, it's also possible for you to inherit a genotype that provides you with the genes necessary to code for both A antigens and B antigens, meaning you'll have both of those on the surface of your red blood cells. And as a consequence, your body will not produce, if you have AB type blood, and therefore you have those antigens on your surface, you will not produce either type of antibody in your circulation. Otherwise, you would have an autoimmune condition, and a fairly significant one at that. Lastly, it's also possible for you to, through inheritance from mom and dad, to not receive either the A gene or the B gene, in which case you won't have either antigen on your surface of your red blood cells. But what your body will do as a consequence is it will produce both antibodies, anti-A and anti-B antibodies. Okay. Now, this has various implications for blood donor and recipient, which we'll talk about more later, but that's kind of the framework of what the blood typing system is. There's also this aspect of positive and negative, which corresponds to the RH slash otherwise known as the D antigen, but we'll talk about that more when we get to our scenario a bit later. Now, here we have what is called a pedigree diagram. All right, so this is a little bit of genetics, so we're going to take a slight detour. For some of you, this is relevant. For others, maybe not so much, at least not right now. So we'll just do a quick, brief summary of this. So if you think back to like high school biology, you probably covered a little bit of genetics with pedigrees and Venn diagrams and all that sort of jazz. So we'll just do a brief intro right here. Now, in this case, there's this concept known as dominance and recessive allele, dominant and recessive alleles. If a gene is dominant, that means that if you have it, you will express it. All right. And if it's recessive, that means you're only going to express that gene in the absence of one of the dominant ones. So in the case of blood typing, with regards to the antigens, A and B are known as codominant genes. All right. Meaning that if you have either or both of those genes in your genome, you will express that phenotype. You will express that antigen. So if we look at child 2, for example, right here. So child 2 received the A gene from their dad, and they received what we'll call an O gene. In other words, a gene that doesn't code for either This is a gross oversimplification, but we don't need to get into the weeds right now. Let's say that this little negative sign is an O gene right here, and they received the O gene from their mother, okay? So in this instance, since A is a dominant gene, they will express that gene. Therefore, they will have A antigens on their surface, and the O basically isn't doing anything in this case. The opposite is true over here with child 3. So child 3 received the B gene from their mom and received an O gene from their dad. Again, B is also codominant with A. So therefore, this patient or this individual will express the B antigen on the surface of their red blood cells, and the O is just kind of hanging out there, all right? And then lastly, because of that codominance, meaning if you have either or both, you'll express both genes. In the case of child 4, this person received the A gene from their parent, from their dad, and they received the B gene from their mom. So therefore, they will express both of those genes simultaneously, hence giving them a blood type of AB with A and B antigens on their surface, on the surface of their red blood cells. Lastly, as an example, the only situation under which you can have an O-type blood, meaning you're not expressing A or B antigens on your surface, is if you don't receive either an A or B from both of your parents. So in this case, for child 1 over here, they received what we'll call an O gene from their father and an O gene from their mother. And since they don't have either A or B genes, they'll not express either of those antigens on their surface, hence giving them the blood type of O. Okay. Again, there's more nuance to it than that, but it's sufficient for right now. All right. Now let's talk about our third category. So if you've heard someone say like, oh, I'm blood type A positive, all right, what does that mean? Well, now we know that the A corresponds with the A antigen, so they're expressing those. But then there's that positive negative at the end of the blood typing. What is that referring to? Well, what that's referring to is this concept known as the Rh antigen or the D antigen, and therefore also the D antibody, okay? So if you're positive, that means you do have a copy, excuse me, you do have a copy of the gene that codes for the D antigen. So therefore, you will be positive. If you're negative, you don't have that gene. Therefore, you don't express it. This has the same dominant inheritance pattern as well as we saw previously with this. So the Rh gene is a dominant gene. So if you have a single copy, you will express it. If you don't have a copy from either of your parents, it's not relevant to you. Now we have this little scenario case down here called hemolytic disease. So this is hemolytic disease of a newborn, and this is the main scenario that we need to understand because it's very clinically relevant for mothers and their children. So imagine, if you will, with me, a scenario where we have a mother who is Rh negative, meaning she doesn't have the gene for the Rh antigen. Therefore, her red blood cells do not express this gene, okay? Now, differently from the whole inheritance pattern over here with A and B blood, we don't have this sort of like if you have it and then you produce antibodies, or if you don't have it, if you don't have it, you produce antibodies. If you do have it, you don't produce antibodies. Not quite the same relationship here. So in this instance, mom does not have the gene, so therefore her red blood cells do not have the Rh antigen on their surface, but their child, because of inheritance from dad, their child is Rh positive. So what does this mean in terms of the pregnancy? Well, during the first pregnancy, we're not going to have an issue because when it comes to blood flow, the blood flow of mom and child are essentially closed loops. They're independent of each other. They never directly interact. The point of interaction between them is just for the purposes of gas exchange within the placenta. But the placenta just allows the vasculature of mom and baby to come in close proximity to each other, very similarly to how the alveoli of the lungs work. So they come right up next to each other, but they never directly interact. But oxygen can diffuse across, and CO2 can diffuse in the other direction. So during first pregnancy, we don't have an issue, okay? Because mom's immune system is not going to interact directly with the Rh positive blood of baby, and therefore won't have an infectious reaction to it. However, when during the process of delivery, during giving birth, the placenta will separate off of the lining of the uterine wall, and there will be some vascular damage that occurs there. And at that point, when the placenta separates from the uterine wall and that vascular damage occurs, there's the chance that mom and baby's blood will interact with each other. And as a result, that Rh positive blood from baby may interact with mom's immune system, which is Rh negative, and it will view those Rh positive antigens as a foreign pathogen. And therefore, it's possible for mom to now start to develop antibodies against the Rh factor, okay, against the Rh antigen. So during first pregnancy, this isn't a problem. But now, for example, let's say that she has a second pregnancy where she has another Rh positive baby. All right, now during the first situation, there were no antibodies in circulation. But now during the second pregnancy, because of that interaction during delivery, mom's immune system is now producing anti-Rh antibodies. Now, whereas the blood flow themselves don't directly interact across the wall of the placenta, antibodies are small enough molecules to where they can cross over. So the antibodies from mom that are anti-Rh will go across the placenta into the baby, and they will start agglutinating the baby's blood, which is a very severe condition. It's called hemolytic disease of a newborn. And this is a very bad thing. And so one of the things that will have to happen, uh, typically, when the mom goes to like her OBGYN doctor, is they'll do a blood test and they'll check what blood type the baby has. And if there is this incompatibility, then they'll basically provide a serum. They'll do an injection with mom. Typically, it happens twice, kind of early on in the pregnancy and then about halfway during the pregnancy, week like 24, if you will. That will essentially inactivate those antibodies, those anti-Rh antibodies in her bloodstream. So that way, when they do cross over, they don't interact with baby's blood and we don't have that process of agglutination. Okay. So that's going to be the Rh antigen and antibody. Now, in summary, just to make sure we're tracking with this, again, if you have type A blood, you have A antigens on your surface, you'll have anti-B antibodies in your circulation. The opposite's true for B type blood. In the case of AB blood, you have both antigens on your surface, therefore you'll have no antibodies in circulation. Otherwise, you would have a severe autoimmune condition. And then lastly, in the case of O type blood, we have neither A nor B antigens on our surface, but as a consequence, our immune system will naturally produce antibodies against both A and B. And then with regards to Rh, which we just talked about, Rh is inherited independently of A and B, so therefore it's its own factor that needs to be evaluated where we have positive and negative type bloods. And however, it can result in that same process of agglutination if we do have an incompatibility. So when you're matching up blood types, it's another factor that you need to consider. And again, we had that example of inheritance, or sorry, not inheritance, but rather interaction between mom and baby during not the first pregnancy, but second pregnancy, where her body would now be producing anti-Rh antibodies, which would negatively affect the baby who's Rh positive if we don't give them an injection, okay, to inactivate those antibodies. Now, in terms of interpreting blood panels, if we're doing a blood typing test, well, how do we do that? Well, we have these little petri dishes that we can use. And so what we'll do is we'll take a couple samples of a patient's blood, and then we'll provide anti-A antibodies and anti-B antibodies to that serum. We'll also do the same thing for Rh or anti-D antibodies, and we'll see where we have a reaction. Now, when it comes to interpreting these things, here's the super simple rule. If you have a reaction, you have that thing. All right. So in this case, for the anti-A antibodies, we do have a reaction right here, which means this patient's blood in these two petri dishes right here, or these two little droplets, if you will, this particular patient does have A antigens on the surface of their red blood cells because the anti-A antibodies interacted with it. So therefore, this individual would have type A blood. The opposite is going to be true here with type B. So notice we don't have a reaction with the anti-A antibodies, meaning this patient does not have A antigens on the surface of their red blood cells. However, we did have a reaction with the anti-B antibodies, meaning they do have so many terms to keep straight. They do have B antigens on the surface of their red blood cells. In the case of AB, we'll see a reaction in both. And in the case of O, since we don't have any antigens on the surface of our red blood cells, we won't react with either of the antibodies, and therefore we'd see no reaction down here. So if you see a reaction in a particular dish, you have that thing. All right, that's the take-home message right there. Similar information right here. Not going to particularly cover that. So now we're going to take a little bit of a practice, detour, and we'll go through a couple examples right here. So I'm going to switch over to my pen, and we'll run through this. And each one of these is basically its own little scenario right here. So this is a particular patient. This is a particular patient. And we'll try to figure out what blood type they have based off where we have a reaction. So as a starting example, let's say we have a reaction right here. We have agglutination with the clumping of the red blood cells right here. And then we have some clumping over here. All right. What type of blood does this patient have? Well, again, wherever we have a reaction, we have that thing. So in this case, we would have sorry, I don't know how to draw very well with the cursor. I play video games with a controller, not with mouse and keyboard, so I'm not very good. So this, it's really not good. So this particular patient would have A positive blood because we reacted with the A antibodies, and we reacted with the anti-RH antibodies right here. So this patient would be A positive. If we just had a reaction right here, okay, but we did not have a reaction with A or RH, then this would be B. I'm trying. This would be B negative blood, okay? Finishing out two quick examples. Let's say we had a reaction right here, and we had a reaction right here. All right, so we're reacting with anti-A antibodies and with anti-B antibodies. That means this patient has A antigens and B antigens on the surface of their red blood cells, but we have no reaction in this dish right here with the anti-RH. So this person would be A, B, oh my gosh, AB negative right here. And then lastly, let's say we had no reaction at all. Let's say this was the final output for this particular patient, okay? What type of blood would this person have? Well, they would have O, oh my gosh, O negative blood, all right? Now, with bearing that in mind, should make sense. Let's talk about compatibilities, because compatibilities can sometimes be a little bit tricky. Now, the way I like to start this off is we'll start off at the extremes and then we'll go to kind of the middle ground, okay? Now, right here, let's go ahead and start off with O negative type blood. So if we have a patient with O negative blood, who can they give or receive blood from? Now, here we have the recipient's blood type. So when we're looking on this side, we're looking at recipient's blood. And here we have the donor's type of blood up here, all right? So if the recipient, if our patient's blood type is O negative, who can they receive blood from? Well, they can only receive blood from another O negative individual. Now, why is that the case? Well, if their blood type is O negative, what that means is, my cursor went away. We'll use the pen right here. All right, if we're talking about a patient who is blood type O negative, well, if this is their red blood cell right here, what they don't have on the surface of their red blood cells is they don't have A antigens or B antigens or RH antigens. But as a consequence of that, what are they going to have in circulation? They're going to have anti-A and anti-B antibodies in their circulation. We also don't want to give them RH because that's going to make them not have a good time, potentially on first interaction or certainly on second interaction. So they have little antibodies. I can't even draw antibodies like this, floating out in their blood plasma like this, okay? So if we were to give them, for example, let's say we gave this patient, we'll do B positive blood, okay? So if we give this patient B positive blood, well, these red blood cells from a donor with B type blood are going to have B antigens on their surface, which are therefore going to interact with the antibodies right here that are anti-B antibodies floating around in our patient's bloodstream. So we would have that process of agglutination, which we want to avoid. And so this would be very bad. We don't want to have this happen. Now, flipping over to the other side, let's say our donor is O negative blood and they're giving blood to someone else. Who can they give blood to? Who can someone with O negative blood give blood to? Well, in this case, they can actually give blood to anyone. And the reason that's the case is because, again, if the donor right here, is O negative type blood and this is their red blood cell, or their red blood cells don't have any markers, they don't have any antigens on their surface that would react with some of the antibodies of the receiving patient's blood type. So therefore, as a donor, they can give blood to quite literally anyone right here. So if they give blood, for example, to a patient that has B type blood right here, well, what does this B blood type individual have? Well, they're going to have red blood cells that have B antigens on their surface. And what they'll also have is anti-A. Oh my gosh. This is so bad. Is the drawing even worth it? I don't know. They're going to have B antigens on their surface. B antigens on their surface, and they're going to have anti-A antibodies in their circulation. Well, since our O-type blood that we see up here doesn't have any antigens on their surface, they're not going to react with the anti-A antibodies in circulation for our receiving blood type of B positive's blood. Okay, you know what? I promised you we were done with that last example. We're going to do one more real quickly. Let's talk about A negative type blood right here. Okay, so as a recipient, A negative can always receive O negative blood because that's the universal donor. Because again, O negative blood doesn't have anything that our immune system would interact with on the surface of its red blood cells. So we can obviously receive that. And of course, we can also always receive blood from our exact same blood type. So therefore, we can receive A negative type blood. However, notice this omission right here. We can't receive A positive blood. Now, why can we, as an A negative individual, not receive A positive type blood? Well, if we draw our red blood cell right here, all right, if this is our A negative blood, what we have on our surface is we have A antigens on our surface. Since we're negative, that means we don't have RH antigens on our surface. So that means we are at risk of developing antibodies against RH upon exposure. Okay. Sometimes this can happen with the first reaction. It certainly will happen with subsequent reactions. Okay. And so, since we don't naturally have RH antigens on our surface of our red blood cells, we are at the risk of developing anti RH antibodies on exposure. Therefore, we should not give this patient A positive blood because this blood type right here would have red blood cells that look like this, where we have the A antigens on our surface, but also this donor's blood has RH antigens on its surface, which we are at risk of interacting with. And this would be very bad. We don't want to expose our blood to this potential immune reaction. Okay. Anything else? No, that's going to be it. Alrighty. See you in the next one. Have a great life.